Thank you all for attending this talk. It's kind of hard to put all the work that I've been involved with into a single box, but the title I've landed on is the physical quantification of the interactions between environment, physiology, and human performance. And if that doesn't make any sense, hopefully it'll make a little bit more sense by the end of the talk. So the starting place of all this is the fact that our environment has a tremendous impact on our bodies and performance. An example of this is the role air quality makes on our health. So there are very well-known health effects, negative health effects of poor air quality, short-term things such as allergies. It's spring, allergy season, so maybe some people are experiencing this right now, coughing, headache, exacerbation of asthma, but also long-term disease such as lung cancer, heart attack, stroke, and dementia and Alzheimer's. According to the World Health Organization, in 2015, about 9 million people died worldwide due to bad air quality. But there's more to the story here because all of us have this thing in our heads called a brain, and poor air has also been associated with cognitive decline, reductions in cognitive performance. So maybe you've seen headlines such as, "Is air pollution making you dumb?" The more scientific version is the role of air pollution in cognitive impairment and decline. But still, air quality, with the significant number of impacts it has on our physiology, is just one aspect of the environment. There's temperature, which can cause heat and cold stress, has a significant impact on our sleep cycles, on our cognitive health and performance, on our ability to thermoregulate and maintain homeostasis. There's light, which allows us to see the world around us, also plays a tremendous role in our sleep cycles, affects our pupil size, our mood, alertness. There's chemosensation, which we experience through smell and taste, but also if some chemical gets on your skin or into your eyes, it irritates you. And then there's also the constant sensing of the partial pressure of oxygen and carbon dioxide in our blood, which is key for all the cells in our body. There's sound, which can grab our attention. There's the emotional context. So not just the physical things that we interact with, but how we interpret and understand these things, and so much more. The goal of this research is to capture this full picture to enable holistic analysis. A lot of work has been done in pursuit of this goal, and then this is just like a brief timeline of some key points of my graduate school education. Some program landmarks are highlighted in red. 9 different publications where I'm either a first author or co-author are shown in green here. 2 different awards are shown in black, awards and honors. And then for the past 6 semesters, we've been mentoring different groups of computer science undergraduates for their senior design projects, and those are shown in blue here. So the way I'm going to outline this talk is I'll start with the motivation, why we want to do this work, and then talk about 2 key ingredients for making this research possible, which are 1, holistic sensing. The goal of holistic sensing is to capture a comprehensive view of a system of interest. And then the second key ingredient is machine learning, which allows us to take this large volume of data that we collect and make sense of it. And then I'll zoom into 3 specific projects that are all connected to this goal. So the motivation of this work, again, we know the environment affects us, but the full story of how the environment impacts our body, our physiology, our performance is far from understood. And part of the problem is the interactions that we're interested in here are very high dimensional. There's a lot of them, and they're interconnected. So we pursue a solution to this problem through 2 key ingredients. The first being holistic sensing, which again is the pursuit of a comprehensive snapshot of a system of interest, and machine learning, which allows us to make sense of this snapshot. This idea of holistic sensing sits at the cornerstone of what we do in our research group. So our research group is called MINTS, Multi-Scale Integrated Interactive Intelligent Sensing and Simulation. All the work that I'm going to be talking about today fits into this larger vision of MINTS. This holistic analysis of the environment is enabled through these 8 different sensing sentinels, which is all the way from global satellite data down to the hyperlocal of robotic boat, a ground survey vehicle, walking robot, and then 24/7 distributed sensor networks. But more recently, we've expanded into the human domain, the biometric domain, and bringing these 2 key pieces together has been my focus for the past few years. And the second key ingredient is machine learning. And this basically allows computers to learn by example. So if you want to do machine learning, there are 2 key requirements. First is a comprehensive data set, which we can achieve through this holistic sensing sensing paradigm, but also you need a machine learning algorithm. Typically what this looks like is you'll have two different types of variables in your data set. You'll have one target variables, which are quantities that you're trying to estimate or predict, and then you also have predictor variables, which are the quantities you use to make that estimation or prediction. What the machine learning algorithm does is it learns the connection between the predictor variables and the target variables. Machine learning can be broken down into two general steps. The first step is training. So here you'll take part of your comprehensive data set to form a training data set. You'll feed this data to a machine learning model, and out of the model comes a machine learning model, essentially a computational representation of the relationship between your predictor variables and your target variables. The second step is prediction. So you'll take new data that your model has never seen before, you'll give it to your model, and it'll spit out an estimation for that new data. I'll be focusing on just 3 studies here, even though there's more to talk about, but all 3 of these studies approach this overarching goal of quantifying the interactions between environment, physiology, and human performance from 3 different angles, and they have a shared experimental paradigm which make use of these 2 key ingredients I was talking about earlier, namely holistic sensing and machine learning. So the first study, this was the first thing I started working on when I started working with Dr. Lari. The title of this work is Modeling Autonomic Pupillary Responses from External Stimuli Using Machine Learning. So basically the goal here is to model the relationship between pupil size and ambient light information. The first key concept here is light. The light that we commonly interact with is typically not made up of a single color. So if anyone is a fan of Pink Floyd or has seen this illustration before, you've seen white light entering a prism and then a rainbow coming out. And what this is depicting is that white light is made up of several constituent frequencies. It's made up of several constituent colors. You can characterize this property of light through its spectrum. There's some different examples of light spectra here. The thing to look at is the x-axes are showing wavelength, which is essentially the color of the light, and then the y-axis is showing the intensity. There's some examples like daylight and sunset, even though they're coming from the same thing, the sun. Since the solar angle is changing throughout the day, what we see here at the surface of Earth changes dramatically over the course of a day. But then you also have artificial light sources such as fluorescent bulbs, incandescent bulbs, and LED bulbs, which have very different light spectra. So you can use this frequency information to characterize different light sources. Then this second key part of this study is how we perceive light. We decode light information. We see light through our eyes. And basically how this works is light enters the eye. It gets focused onto the retina, which is essentially a stack of cells. And a subset of these cells are photosensitive. So basically they respond and change when presented with light. There are 2 general types of, well, there's more, but 2 I'll talk about here. Photosensitive cells. The first are rods, which are involved in these low light vision or scotopic vision. And then there are also cones, which allow us to see color. They capture frequency information. And then of these cones, there are actually 3 different types. There's a type of cone that's more sensitive to blue light, so-called shortwave photoreceptors. There's another more sensitive to green light, and then another class more sensitive to greenish yellow light. That's what is being shown here. This is just the response curves, the sensitivity curves of these 3 classes of photoreceptors, which we'll come back to later on in the talk. Another key part of this story are the pupils, which are the dark part of the eye. This is what controls the amount of light that is shown onto the retina. The pupils actually change size, and when they increase in size, it's called pupil dilation. When they decrease in size, it's called pupil constriction. And this is largely an autonomic response, meaning it happens automatically. Most people can't control the size of their pupil on demand. It's typically something that can be indirectly affected through different means. There are a few different things that can modulate pupil size. The first is light. As you go into a dark room, if you could look at your pupils, they would actually increase in size. And if you were to walk outside, your pupils would actually shut down and get as small as possible, presumably to protect your retina. You don't want to burn out your retinas. The second thing that can modulate pupil size is actually cognitive load, exerting mental effort, so to speak. This is like trying to solve the math problem, what's 81 times 17? If someone were to look at your pupil while you're thinking and try to figure that out, they would actually see your pupils increase in size. Another thing that modulates pupil size is viewing distance. When you switch from looking at something far away to something that's close to you, your pupils will tend to constrict. In this study, we're directly measuring light, and then in some sense, we're indirectly measuring these other things, which we'll get into later. So the way we're capturing this information, one, information about our environment or the ambient light, and then information about the pupils is through these two sensing systems. In this work, data is collected during 3 outdoor walks for a single participant, which was actually me. And then this is a machine learning pipeline. So we have our input variables or predictor variables. This gets fed into a machine learning model that we develop and train here. This actually uses an ensemble of decision trees for regression. I won't get into all the details of that, but I'll just say that for the aficionados out there. And then the outputs are actually 3 different targets, which generate 3 separate models. And those are the left pupil diameter, right pupil diameter, and then the average of the two. Before diving into the machine learning, we'll just take a look at this and get a sense of what's being measured here. The way to look at this is this top colorful plot is showing the light spectra over time, going back to that earlier discussion about light and being made up of different colors. The x-axis is showing time. So as you move along the x-axis, time is moving forward. The y-axis is showing wavelength, or essentially the color of the light. And the color that you're seeing in this plot is representing the intensity of the light. Dark red is a very intense color, and dark blue is a very low intensity. What's interesting is this walk was both indoor and outdoor, so you can tell when it's outdoor because you see that characteristic peak in the blue light that we saw earlier for sunrise. And then you can tell when I walk inside because of the fluorescent bulbs that were in the lab we were working out of. So you can see this characteristic shift in the light spectrum. At the bottom here, we again have the x-axis showing time, and then the y-axis is showing the left and right pupil diameters measured in millimeters. So you can see whenever I was inside, the pupil diameter is pretty large. It's around 4 millimeters, but right when I go outside, it rapidly decreases to just above 2 to 3 millimeters, and then periodically will spike up, and that's presumably for walking into shaded regions outside. And then right when I come inside, the pupils rapidly start to increase in size, and they'll respond to even the subtle changes in light spectrum inside the lab we were working out of. Moving on to the machine learning. So this has been done before, but typically people had just used a single predictor variable. They would just use total light intensity or some proxy of that to try to predict pupil size. But here we use not only that, but also the full spectrum of visible light in addition to the orientation of the head. The way to look at this plot is we have the estimated pupil size on the x-axis here, and then the true pupil size on the y-axis here. So what that means is when the estimated and the true pupil size are exactly the same, those values will sit on this y equals x, this one-to-one black line here. And what that means is it's a perfect estimation. And then anything that deviates from this one-to-one line is not a good estimation. All these different colors and dots you're seeing in the background here, they are the predictions of past models. For example, this past model might tell you the pupils are just above 2 millimeters. That would be the model's estimation, but the true pupil size could be anywhere between 2 millimeters all the way up to like 6.5 millimeters. There's a very large variation here. But if you're looking at the red and green circles and pluses here, that's the performance of the machine learning taking into account the full spectrum of visible light. We didn't just do this for average pupil diameter, we actually did this for the left pupil diameter, right pupil diameter, and then the average of the two. So we have the same kind of plot. It might be kind of small, but in the top left corner here, we have the estimated on the x-axis, the true on the y, and then anything sitting on this black line is a perfect estimation. The green circles here are showing the data from our training data set. So basically, that's the data we used to develop the machine learning model. And then these red pluses are data from a testing or validation data set. So that's data the model has never seen before. So you can see we can quantify how well the estimated and the true pupil size agree with each other through an R-squared score. The training data set had an R-squared of 0.99, which is really good because the best R-squared you can have is 1, which means a perfect fit. And then the validation is 0.96. So the machine learning basically nails the prediction of this very complicated interaction. And then it's the same thing for the right pupil diameter and then the average of the two. Those actually had better performance than the left pupil diameter model. What's really nice about this machine learning approach that we used is not only do you get an empirical model of some complicated interaction, but you also get a predictor importance ranking. So the model will tell you which predictor variables are the most helpful in making the estimation. So if we kind of look at the top 3 predictors across these 3 models, we can see numbers like 567 nanometers, 568 nanometers, 563, 562, a lot of predictor variables around 560 or so nanometers. And if we kind of look back to this mean absorbance curve from before, this lines up with the long wave photoreceptors that we have in our eyes. So this long wave photoreceptor is most sensitive to these colors that we're measuring. And so it kind of lines up with the underlying physics physiology. There was a major drawback in this preliminary work, namely that we had to drop a significant amount of data because the eye tracking glasses don't operate well in very bright ambient light conditions. Reason being is the eye tracking glasses actually use infrared light to track where the pupil is and ultimately estimate the pupil size. So when you go outside and there's the bright sunshine and a lot of infrared light outside, it'll actually wash out these infrared light sources that the glasses use to make the estimation. So basically any data record that was outside the sensing range of the eye tracking glasses, we just dropped from the analysis. Some conclusions from this work is we use this holistic sensing and machine learning based experimental paradigm to derive high fidelity, high accuracy empirical models of pupil diameter using spectral light information. And this model outperformed all previous models that used just light intensity alone. And then the top predictor variables were the spectral irradiances around 528 and 565 nanometers, basically the light intensity of colors around those wavelengths. Those correspond with the mean absorbance peaks of these middle and long wave photoreceptors in the retina. For future studies, we need to make sure not to use the eye tracking glasses in environments that are too bright. Also, we want to expand to more participants and expand the number of environmental and biometric variables. So that's a perfect segue to the next study where we actually do this. We expand the data collection to 5 participants and we expand both the biometric and environmental variables that we're measuring. The title of this study is Dense Urban Environment Dosimetry for Actionable Information and Recording Exposure, or DUDAIR for short. Even though we have data for the 5 participants and we have all these different environmental factors that we measured, here we're just going to focus on a single measure, which is particulate matter, in other words, air quality, because of its tremendous societal relevance and impact. Also, we focus on a single participant for numerous reasons, but one of the key ones is to avoid the inter-participant variabilities and just look at a single participant. Air quality is how I opened up this whole talk. According to the World Health Organization, we have scary statistics. In 2016, 4 million premature deaths worldwide due to bad air quality. In 2019, 99% of the world's population lived in areas that fell short of the WHO's air quality guidelines. The main bad actor, the main constituent of bad air is particulate matter. And that's just basically really small bits of matter that are suspended in the air. There are 2 broad classifications of particulate matter. There's the so-called PM10, which is all particles up to a size of 10 micrometers in diameter. And then there's PM 2.5, which is the same kind of story, but it's all particulates up to 2.5 micrometers in diameter. Here's a little cartoon in the top right corner here. That giant yellow circle is the diameter of a human hair. The green circle is PM 10, and then that little orange circle is PM 2.5. Here's some different examples. So again, we have human hair for scale on the far right-hand side. To the left of that, we have a grain of sand. To the left of that, we have a grain of salt, and so on. When we're talking about PM10, we could be talking about dust. That's relatively close to the size of a red blood cell, respiratory droplets. And then when we're talking about PM 2.5, we could be talking about dust, but we could also be talking about smoke, bacteria, and then the really small stuff we have viruses, such as the coronavirus or Zika virus. The other key part of this story are these autonomic responses, which I kind of touched on earlier with pupil size. Again, these autonomic responses are these automatic things that happen. They're automatic physiological mechanisms meant to keep us safe. And we can actually capture these automatic mechanisms through the biometric sensing. Here are a few examples of autonomic responses. There's pupil size or pupil dilation, which I talked about earlier, but there's also heart rate changes. Let's say you go for a run or start doing some jumping jacks, your heart rate's going to increase. You don't have to deliberately decide to increase your heart rate. You can decide to exercise, but your heart rate's going to increase whether you like it or not. There's also sweating. Most people I know can't control whether they're sweating or not on demand. The key thing about these autonomic responses is one, they happen automatically, but they also happen very rapidly. Here's the little video of me wearing the eye tracking glasses, and then this is a video of my eyes. If you kind of lean in and look at my pupils, you can see them constantly dilating and constricting in real time. This is data of me reading a Twitter feed, I think it's reading my Twitter feed. So you can see how sensitive your pupils actually are. This presents an issue. The current research of air quality and its effect on our health is typically done on the epidemiological scale. So that's on the scale of cities and with a time resolution over the course of days. But these autonomic responses happen very rapidly, and this is on the scale of us, which are the scale of meters, not kilometers, and over the time scale of seconds or even faster, milliseconds. So there's an issue of scales with a lot of the work that's readily available out there. Our solution to this is to investigate the impacts of particulate matter on ultrafine spatial and temporal scales. We achieve this through the same experimental paradigm that I was talking about before. We bring together the holistic biometric and environmental sensing. We feed that into a machine learning pipeline, and then from that machine learning pipeline, we can evaluate the key associations between the biometric and environmental variables. Here are some real pictures. On the left here, we have the backpack, which houses all the computer, the recording unit, the biometric sensors. On the right, we have a picture of the trunk of our environmental survey vehicle, which has all the environmental sensors in it. And then here in the top right, our first participant was actually Dr. Laurie, and we collected data at Breckenridge Park. That's him riding along in the electric survey vehicle, following safely behind. So we capture these autonomic responses through biometric sensing, and they provide a window into our underlying physiological and cognitive processes. So again, here we're using the eye tracking glasses, but we're sensing a lot more than just the eyes here. We sense brain activity through a 64 electrode EEG. We're looking at the blood oxygen ratio, heart rate, the sweat response through galvanic skin response or GSR. We're looking at body temperature. We're looking at the heart activity with ECG. And then for the environmental side, this is the whole suite of environmental sensors we have in the survey vehicle. But again, because of its societal relevance here, we're just going to focus on particulate matter, which I was talking about earlier. And we use this reference sensor to measure the particulates. So it's the same kind of story. We have our predictor variables, which are the biometrics, and then we have our target variables, which are the PM concentrations. And the idea here is if we can use biometrics to accurately predict particulate matter, then that's an indication to us that particulate matter is impacting these biometric variables. This combination leads to the generation of 6 different machine learning models corresponding to 6 different particulate matter targets. And then once we have the machine learning models, we can generate these 2 key data products. One, we can evaluate the performance of each model, but then we can also evaluate the importance of each predictor for every one of the models. Here's a more comprehensive view. This is all the biometric inputs on the left here. Again, we're using the same exact machine learning algorithm, the ensemble of decision trees for regression, and then we have 6 different particulate matter related variables here. We also use this Occam's razor principle in this work. We have these 329 biometric predictor variables, but we have our friend Occam here who says simpler models are better. So he comes up with his razor and reduces it to the 9 most important predictor variables. So we go from this very large number of variables that it's very difficult to interpret and just focus on the most important 9. Even though we have data for 5 participants, this work I will be talking about analysis from 1. There are different kinds of data collections that we do. We do baselines with the eyes open and closed to get someone's baseline biometric values. And then we also have a ride to this park, and then while we're at the park, we'll ride around in circles collecting all the data. The data I'll talk about here was actually collected over the course of 3 separate days and 7 trials across those 3 separate days. Here's a video. This is Dr. Laurie riding the bike with all the biometrics, and then this is the survey vehicle full of the crack team of scientists. So we're going to zoom in into one of the models, and then we'll take a step back and compare all the different models together. The model I'll talk about here is the one for PM1. So just like there was PM10, all particles up to a size of 10 micrometers in diameter, PM2.5, and then there's also PM1, which is particles up to 1 micrometer in diameter. And this is measured in micrograms per cubic meter. I'll direct your attention to this top left plot here. So it's the same kind of plot we saw earlier with the estimated target value on the x-axis, the true target value on the y-axis. You have a good fit if all the data points are sitting on this black line, this y equals x line. Again, we have our training data set, which is shown as blue circles, the validation data set shown as red pluses, and the machine learning does a great job at capturing this relationship between these 9 top biometric predictors and PM1. And then off each axis is actually the distribution of the estimated PM1 values and the true PM1 values for the training and validation data sets. Another way to compare the distributions of the estimated and true PM1 is through this quantile plot, which is essentially plotting the different percentiles of the estimated and true PM1 values. At the bottom here, we have a time series showing the true PM1 values in black, and then the machine learning model estimated PM1 values as a dashed red line. So you can kind of see that the machine learning does a pretty good job at doing this estimation, since the red line is pretty close to this black line. Again, this data is over over the course of 3 separate days for 7 trials. And then this time index is showing every 30 seconds. There are these discontinuities between trials because there's some changeover or it's a completely different day. Just like before, we had the predictor importance ranking. We have the same kind of thing here. For estimating PM1, we actually found that temperature was the most helpful biometric predictor in making this estimation. We'll come back to that. So we'll just take a step back. That was just one model. Like I was saying earlier, we had 6 different particulate matter PM targets, which translated to 6 separate models. What we have here is a plot showing the relative accuracies of each of the machine learning models. So on the far left, we have PM1, which we kind of dove into the details of. And to the right of that, we have the model for PM2.5, which was the next best performing. And then PM4 to the right of that. And then DCN. And what DCN is the particle count density. So you take a volume of 1 meter and you just count all the particles in that volume, and you get the count density. To the right of that is PM total, which is basically the sum of PM1, PM2.5, PM4, and PM10. And then the worst performing model was for PM10. So this leads to one of the key observations of this work, which is that the smaller particles were better estimated than the larger ones. There are 2 potential explanations for this that we came up with. One, there are far fewer larger particles than smaller ones, which leads to them being not as well mixed as the smaller ones. So there's less of an agreement between what the participant is breathing and what is sampled by the survey vehicle just a few meters away. The second explanation is that the larger particles have less of an impact on our physiology. And this kind of lines up with what is known about the penetration depth of different particulates. Namely, the larger particles like PM4 and PM10 will only make it to the nasal cavity or maybe the trachea, but the smaller particles like PM2.5 will make it to the upper part of the lungs, and the PM1 will actually make it to the alveoli, which are these small sacs that enable our bodies to dump off CO2 and take in oxygen. Just like we had a predictor importance ranking for a single model, we can do an aggregated predictor importance ranking. So this isn't just for a single one of the models, this is for all 6 models. And again, we see body temperature is the most important predictor variable in making these PM concentration estimations. Going back to this observation that the smaller particles are better estimated, to kind of look at this more closely, we didn't just want to look at these broad particulate size ranges of PM1, PM2.5, PM4, and PM10, but a much finer size resolution. What we have here on the x-axis, we have 40 different models ranging from particles that are 0.18 micrometers in diameter all the way to 10 micrometers in diameter. So you can see as the size increases going from left to right, you can see the model accuracy of these 40 different machine learning models drops significantly, especially around this 2 to 3 micrometer size range. Again, this lines up with what is known about the penetration depth of different particle sizes. The largest ones above PM2.5 are inhalable. They'll make it to the nasal cavity and trachea. Particles between 1 micrometer to 2.5 micrometers are called thoracic because they'll make it to the upper part of the lungs. And then the smallest particles are called respirable because they'll make it all the way down to the alveoli and even into the bloodstream. Conclusions of this work is we use the same exact experimental paradigm as before, but now in a completely different context of modeling the relationship between biometrics and PM concentrations. And then we found that we could accurately estimate PM concentrations from the biometrics on these ultrafine scales, which is suggestive of the rapid impact PM has on our physiology. Another key observation was that the smaller particles were better estimated with the biometrics, which had 2 potential contributing factors. 1, the smaller particles were more plentiful and better mixed, or 2, that the smaller particles penetrate more deeply into the human body and have more of an impact. For future studies, we have data for these 5 participants, and we have data for these other environmental variables. It's just a matter of doing the analysis and work, which is something other students are going to be working on in the future. So we're going to switch gears here a bit and put the environmental side of things aside, and we're just going to focus on the interactions between physiology and human performance. In this 3rd study, it's called the Troisgate Pilot Study, and basically here we're evaluating the physiological performance predictors in high-stress live fire scenarios. So basically the goal is to model physiological impacts on human performance in high-stress situations. So the way we generate this high stress is through this training system called Troisgate, and this is a hyper-realistic training environment that places a participant in a face-to-face deadly conflict with an adversary. So in this picture here, we have not a participant, someone actually putting on the study with us. He's kitted up with all the biometrics, looks, he has a real gun in his hand, loaded with real bullets, and he's about to enter this scenario room. I'm about to show a video of him doing the scenario, and then on the sides, you can see his biometrics responding in real time. So that's what we have here. He's entering the room. So that is the actor in front of him. Again, real person. So the actor sees him. He responds emotionally. He yells at him, points his finger at him, grabs a shotgun and shoots him. And then the participant responds. And now you can see his heart rate is starting to spike. The sweat response is elevated. And then you can see his pupils responding as well. So the data collection here, we had 9 participants, which translated to 28 different samples. Again, we do baseline data collections here, and then we do these high-stress scenarios like in the video I just showed. Each scenario is made up of 2 impact points. This is the timeline of the data collection at the bottom here. This picture is me and not a participant in the study. This was a visitor. He was actually now retired, Command Sergeant Major over all of SOCOM, which is Special Operations Command. So that's all the special forces across all different branches of the military. He was part of the leadership there, and he was really impressed with what we were doing. Everyone we were interacting with there that was involved in this human performance research was saying they'd never seen anything like this, bringing together the hyper-realistic training scenarios with the comprehensive biometrics and the expert evaluation of the participants' performance. So again, the machine learning model development is the same story here. We have 329 biometric predictors. We have 27 different performance targets, and this translates to 27 different machine learning models. And again, each machine learning model, we can evaluate its performance and get the most important predictors for each model. Here's a more comprehensive view. I know it's a lot. Key point here is we're using the same machine learning algorithm, an ensemble of decision trees. We have the same 329 biometric variables, and we have 27 target variables corresponding to participant performance. So it's a little bit of math here, but I'll just kind of walk through this. There are 8 performance dimensions listed here, and then you can take all 8 of those dimensions to get the total, which makes 9 dimensions. And then we have 2 expert evaluators, so that's 9 times 2. And then you can take the average of the 2 evaluator scores across all 9 of these different dimensions, and that is how you get 27. So if anyone is curious where this 27 is coming from, that's where it is. This is a similar kind of thing as we saw before. So we're just comparing 11 different machine learning models. They're ordered in descending order of their performance, where the best performing machine learning models are on the left, the worst are on the right. And we see that this performance dimension called cognitive shifting was best reproduced using the biometric variables. Cognitive shifting, we didn't develop the performance dimensions. This was by the group of people in charge of the human performance side, but they defined it as the ability to switch between tactical strategies, such as using an empathetic or compassionate tone and then rapidly switching to like a strong verbal command. The next human performance dimension that was best estimated with the biometrics was this rationality, and then the next best was this self-assessment variable. So we can again do this aggregated predictor importance ranking. So the top 2 biometric predictors of performance were these 2 EEG or brain-related biometrics, which I'll discuss right now. So the first was the baseline scaled beta band power for the AF4 electrode. So it's a lot here, so I'm going to just go step by step. Baseline scaled, basically, since we had 9 participants, everyone has different baselines. So we scaled all the different biometric variables to each participant's baseline recordings. Beta band means a certain frequency of EEG, of brain activation. These are frequencies between 13 and 25 hertz. So a way to understand this is just like light is made up of constituent frequencies or colors, this EEG signal that we measure from the scalp is made up of constituent frequencies or colors. So you can say the beta band is more of a blue if you're considering the rainbow. The last bit of this is the AF4 electrode, which sits at the front right of the head here. And then if you look at this diagram on the right, it's that yellow filled in circle. Okay, so that was a lot. The AF4 electrode sits above this Brodmann area 9, and the Brodmann areas are just classifications of neurons based on their cell type. Brodmann area 9 has been associated with attributing attention, recognizing the emotion of others, and planning. So already you can sense that these types of skills would be critical in navigating these high stress scenarios where it's not always the right decision to shoot. Sometimes you need to empathize and negotiate and de-escalate the situation. A similar kind of thing here. So the next most important biometric biometric predictor was the baseline scaled gamma band power for the P4 electrode. Again, baseline scaled in the same way. Gamma band is just a higher frequency range of activation. So you can think of this as the purple of EEG activity. And then the PF4 electrode actually sits near the back right of the head. This sits over Brodmann area 9. And even though there seems to be less work on this Brodmann area, it's been associated with overriding automatic reactions, attention and spatial cognition, reasoning, and social cognition. So we can dive a little deeper and look at the correlations between these top biometric predictors and the performance dimensions that were best estimated with the biometrics. Here we have the beta band for the AF4 electrode and the gamma band for the P4 electrode. Looking at these plots, you can see that there's a positive correlation. So in other words, as activity in these brain areas recorded from these electrodes increased, performance tended to improve along these three performance dimensions. The third most important biometric was actually the heart rate, and this had a negative correlation with these performance dimensions. So in other words, as heart rate increased away from baseline values, performance tended to drop. Just some conclusions here. We used the same exact experimental paradigm we used in the last two studies in a completely different context of evaluating the relationship between human performance and biometric recordings. From what we're aware of and everyone we've interacted with, it really feels like this was a first of its kind study. Some key correlations were as activity in the AF4 beta band and the P4 gamma band powers, as those increased away from baseline, performance tended to improve, while when heart rate elevated away from baseline, performance tended to decrease. And then understanding these key biometric predictors of performance is important because it can lead to performance improving interventions and ultimately saving human lives, because this could be used for special forces, it could be used for first responders, it could be used for police, doctors, so this can have far-reaching implications. For future studies, we really would want to do more participants than just 9. Also, an idea is to record the biometrics of the actors in addition to the participants to kind of see the relationship, if any, between these two people engaging in this high-stress situation. So just wrapping up, this work has led to a number of contributions. Again, 9 publications where I'm either first author or co-author. We've contributed a lot to open source software, so all the software we write is made open source. All the data is made open access where possible. Given a lot of talks and lectures while in grad school, so here's just a list of those. I've also been making online content, writing blogs on Medium, making YouTube videos, and writing example codes for other graduate students, mainly trying to navigate these ideas of data science and machine learning. I'd like to acknowledge all the funders and organizations we've collaborated with over the past few years. Also, by no stretch of the imagination is this the result of me working in isolation. This is really the result of a large team of people all working in the same direction. And so I would like to gratefully acknowledge and thank all the people I've worked with for the past few years. And then I'd like to thank everyone for taking time out of their Mondays to listen to me ramble on about this. And I'll be happy to answer any questions if there are any.